Hello. In today's video, we shall learn in detail about recombinant DNA technology. By the end of this video, you will have a clear understanding about the steps involved in recombinant DNA technology and its application for human welfare. Let us first understand the term biotechnology. Biotechnology is a branch of biology which deals with the technique of using living organisms, enzymes, or biological processes or molecular biology to produce products for the welfare of mankind. It involves DNA manipulation, that is, recombinant DNA technology, tissue culture, protoplast fusion, etc. Recombinant DNA technology is a technique of manipulating the genome of a cell or organism or bringing together DNA molecules from different organisms or species and inserting it into a host so as to change the phenotype desirably for the human welfare. This results in the formation of new genetic combination. The technique of recombinant DNA was developed in 1973 by Boer and Cohen. It is also known as genetic engineering. Let us first look into the steps involved in recombinant DNA technology. First, isolating genetic material. Second, restriction enzyme digestion. Third, using PCR for amplification. Fourth, ligation of DNA molecules. Fifth, inserting the recombinant DNA into a host. And finally, isolation of recombinant cells. First, let us start with isolation of genetic material. The first step in recombinant DNA technology is to isolate the desired DNA in its pure form with the help of various enzymes. DNA must be free from other macromolecules like proteins, enzymes, etc. The cell or organism from which the required gene is taken is called donor. Next step is restriction enzyme digestion. Restriction enzymes, namely restriction endonucleases, are used to cut DNA at specific locations. These enzymes are called biological or molecular scissors. Restriction enzyme digestion are performed by incubating purified DNA molecules with the restriction enzyme. This results in the fragmentation of the DNA molecule. By the way, if you're getting some value out of this video, please like and share the video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Let us continue with the steps involved in recombinant DNA technology. Third step is amplification using PCR. PCR stands for Polymerase chain reaction. Gene amplification can be done artificially using polymerase chain reaction technique. It is a scientific method of making multiple copies of a DNA sequence using the enzyme DNA polymerase in vitro. It was first developed in 1983 by Carl Mullis. The basic requirements of a PCR technique are DNA template, double-stranded DNA to be amplified, primers. They are oligonucleotides complementary to the sequence present on the desired DNA segment, 4 types of deoxyribonucleotides, namely deoxyadenosine triphosphate, deoxycytidine triphosphate, deoxyguanosine triphosphate, and deoxythymidine triphosphate. They are collectively known as deoxynucleoside triphosphate. A thermostable DNA polymerase that can withstand the protein denaturing condition up to 94 degrees Celsius. Commonly used enzymes are Taq polymerase, obtained from bacteria Thermus aquaticus, and Wnt polymerase, obtained from Thermococcus. PCR methods are thermal cycling, that is, alternately heating and cooling the PCR sample. The 3 essential steps of PCR technique are heat denaturation, heating of DNA at about 91 degrees Celsius. This breaks the hydrogen bond. The double-stranded DNA is changed to single-stranded DNA. Annealing, pairing of primers to the single-stranded DNA segment takes place at about 55 degrees Celsius. PCR primers are short fragments of single-stranded DNA that are complementary to DNA sequences. Primers are also known as oligonucleotides. Polymerisation. Temperature is raised to 72 degrees Celsius. The Taq polymerase adds deoxynucleoside triphosphates behind the primer on the single-stranded DNA. This is one cycle of the reaction. A chain reaction is set up and DNA template is amplified. It is then ligated with a cut vector. Let us understand the meaning of certain terms. First, vectors. Vectors are the DNA molecules used to transfer genetic material into another cell. It may be a plasmid, or a bacteriophage DNA, or artificial DNA or cosmid. The most commonly used vectors are plasmids, viruses, and yeast cells. Plasmids are the small extrachromosomal double-stranded circular form of DNA that originated from bacteria and can replicate autonomously. They are easily integrated with host chromosome. A bacteriophage is a virus that infects bacteria. Cosmids are plasmids having fragments of lambda-phage vectors. Fourth step in recombinant DNA technology is ligation. The purified DNA and the vectors are cut with the same restriction enzyme. Both are ligated by mixing vector DNA, gene of interest, and enzyme DNA ligase to form the recombinant DNA. Enzyme DNA ligase is used to covalently link the two strands into a molecule of recombinant DNA. Fifth step is insertion of recombinant DNA into host. It is the introduction of recombinant vector into a competent host cell. Insertion of a vector into the target cell is called transformation for bacterial cells, transfection for eukaryotic cells, and insertion of a viral vector is called transduction. Final step is isolation of recombinant cells. Since both transformed and non-transformed host cells are produced, marker groups of plasmid vector is used to isolate the transformed cells or recombinant cells. A selectable marker is a gene inserted into a cell which confers a trait appropriate for artificial selection. Let us try to understand the different steps in recombinant DNA technology for the production of human insulin. Human DNA is removed from human cell and is cut with the restriction enzyme. A plasmid from E. coli is used as a vector. This is also cut with the restriction enzyme. The gene for insulin taken from human cell is inserted into the vector DNA. The vector carrying gene for insulin is introduced into the E. coli. The E. coli acts as the host and multiplication of transformed E. coli takes place as shown in the figure. With repeated multiplication, a population of insulin-producing E. coli bacteria are produced. In this way, genetic engineering is used to insert human insulin-producing gene in E. coli. Next, application of recombinant DNA technology. It is used to produce human insulin, humulin, for the treatment of diabetic patients. It is also used to produce human blood clotting factor 8 to treat hemophiliacs. To produce recombinant vaccines for the treatment of human and animal viral diseases. It is also used to produce human growth hormone to treat pituitary dwarfism. It is used to produce bovine growth hormone to increase dairy yields. Also used to produce genetically engineered crop plants or transgenic plants carrying desirable traits like disease resistance, insect resistance, etc. It is used to enhance nitrogen fixing ability and photosynthetic efficiency. It is used to produce human follicle stimulating hormone FSH in vitro. Recombinant DNA technology is also used as a tool of gene therapy to cure or prevent various diseases. This technology is used to treat diseases and improve health conditions by developing new vaccines and pharmaceuticals. Genetically modified microbes are used in biomining and bioremediation. This technology is used to tackle environmental issues such as cleaning oil spills, formation of biofuels from waste, etc. Recombinant DNA technology is effective in getting rid of arsenic particles which contaminate the soil. This technology has wide application in food industries to enhance the nutrient value. So today we have learned in detail about recombinant DNA technology, its process, and various applications. I have some practice questions for you. Please share your answers in the comments section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.